Hello, everyone, and welcome. Today, we're going to be talking about functions of activated helper T cells. This is actually take two of this presentation because the first time I did it, it ended up being like an hour, and who has an hour to spare these days? So hopefully I go on less tangents this time, so it's not an hour. And also, it's not September 2021, although it feels like yesterday. It's like almost halfway through 2022 right now. So just now I'm recording though, so I'm trying, guys. So where we left off last time, activation is reached, right? But just to backtrack completely. So just like any blood cell, right? T cells are born in the bone marrow, right? And then T cells actually mature though in the thymus. There they're double positive. They have both CD4 and CD8 service marker. In the thymus, it'll commit to one lineage. It'll be CD4, which is a helper, or CD8, a killer. Okay, and then it's going to go through some testing in the thymus, and at which point it will basically the thymus make sure that no self-reactive T cells are put out into the lymphatics. So if these tests, if you will, by the thymus are passed, then the T cells, naive CD4 and CD8, can enter the lymphatic system. However, they're not activated because they have not met their cognate antigen in nature. And you really don't want them to, because that would mean we have an infection, right? Or to some extent, right? But yeah, you have these naive CD4 and naive CD8. And then in the lymph node, like paracortex area, they're going to meet a dendritic cell who's basically sampling antigens from the outside world and then gulping up everything around it and then swimming to the nearest lymph node, which takes about a day, as we established last time. And then, so on the dendritic cell, who can do MHC1 and 2 presentation, therefore it can present to both CD4 and 8. Or if I'm going in that order, CD8 and 1, because MHC class 1 is for CD8 presentation, MHC class 2 is for CD4 presentation. And then cognate antigen is found, and then you get like all the co-stimulatory signals needed. So we're, again, just to review, maybe this is why it's going for a while, because I'm doing so many review tangents. You're gonna get T cell receptor. It's gonna, let's just say we have, T cell receptor. It's gonna bind cognate antigen, and the cognate antigen has to be on MHC molecule. The MHC molecule has to be of the appropriate class. So if we have a CD4 T cell, the antigen has to not only be complementary, but for CD4 has to be on MHC class 2. If it was CD8, it has to be on MHC class 1. In addition to this, we have the other co-stimulatory signals needed. You had the B7 on the antigen presenting cell, also known as CD80 and CD86. Lately, I've been saying B7, but this is just different names for the same thing. But it's B7 on antigen presenting cell to CD28 on the T cell. And so these are signals, co-stimulatory signals needed to ensure that there's the okay signal for the naive cell to become an effector. And so if it's an effector, CD8, we established what some of the functions are last time, and we can talk more about them later. And if it's CD4, it'll become an effector CD4 cell. But within that, there is subtypes of CD4. And so that's the basis of today's lecture, the subtypes of CD4. And so these are going to be denoted as Th1. So Th is T helper, right? So Th1, Th2, Th17, and there's also Th0, and there's also T regulatory. So we're going to talk most about the first three, Th1, 2, and 17. And also remember for the effectors, for the effector CD8, there's two pathways in which it can have the activation to become active, to become effector. And the 3-cell mechanism in which it's naive CD8, in addition to dendritic cell, in addition to a helper T cell facilitating the reaction, right? This is your most potent T cells are going to be produced this way. That being said, because CD4s are needed for this process, it takes a while to kick in. So if there's no CD4s to help facilitate the interaction in the beginning, you can actually have a 2-cell mechanism in which it's only dendritic cell and naive CD8 interacts to form the, to form the basis of the effector T cell formation. But these CD8 cells formed from the 2-cell mechanism are not as potent. And just like that, I summarized the entirety of the last lecture in a side tangent. But again, the big focus for today, we're going to be talking about effector CD4 cells and what goes into that. Okay. And we already harped on this. Oh, and also one of the costimulatory signals that I mentioned in the little tangent, also CD40L on the T cell to CD40 on the antigen presenting cell. And again, antigen presenting cell can be dendritic cell, can be macrophage, can be B cell. B cell the most potent, but B cell, because again, just like having the CD4 to help in the three cell mechanism, B cell is also adaptive, so it takes a while to kick in. You won't get B cells presenting antigen until pretty late into an infection, but once they do, it's like overkill. Like they're the best at doing it, right? So what's the key point though about talking about us having subclasses of CD4 cells? Like what does that even mean? So remember, just for the basis, dendritic cell is out in the periphery. Find something red flag, right? Maybe with its TLRs or some other pattern recognition receptor. Okay, so know something's fishy because we're evolutionary programmed to know something is molecularly fishy. That's just the nature of the innate immune system and how really everything starts off with a toll-like receptor. So dendritic cell knows something is something's up. It's going to gulp up its surroundings, and chances are when it gulps up its surrounding, it's going to gulp up antigens in that surrounding, right? Probably even triggered some of the red flags to begin with, right? And so once it gulps up everything, it's going to project everything on its surface as it travels to nearest lymph node, which is a journey that takes about a day again. And again, like I said about that one time I was preparing my lecture presentation on the way to the lecture, like at a conference, same thing. So as the dendritic cell is traveling, it's preparing its presentation. All right, it's very last minute, or I don't know, maybe it has a lot going on in life. It's not for us to judge the dendritic cell. We'd be dead without them anyways, right? So yeah, so on its MHC1 and 2, because remember it can do both, it's going to project what it found. And then it's going to show up in the lymph node, going to go to the paracortex, which is the T cell area, and it's going to project these antigen to the T cells, and the T cells are naive. So again, it's a random, not really random process, but again, in theory, we have T cell receptors to antigen that might not even exist, but we also have some for some that do exist. And so in the chance that the naive T cell finds its cognate antigen, finds its puzzle piece, if you will, you have the basis of being able to recognize MHC reaction, being able to bind respectively T cell receptor to antigen, CD40L to CD40, CD28 to B7, all these things. That's going to happen. But in addition to this, that will make it activator. It'll make it into effector, right? But also there's additional information conveyed from the dendritic cell to the, in the case CD4. Is also, the information is usually in the form of cytokines. It's like a dossier, if you will. And this is just additional information, in addition just to the antigen, about what is the nature of the antigen, or what is the nature of where this antigen was found. So you can almost think of it as the dendritic cell will smother the T cell in like a chemical, and that chemical will direct a certain pattern of growth, right? This is the whole point of this type of dendritic cell dossier, if you will, right? And again, I made this point earlier, and I've really noticed it. Like in the United States, there's medical licensing exams called USMLE and COMLEX. I've noticed that if someone is studying for these exams, in the immunology section specifically, they tend to refer to B7 as CD80 and 86, which is the same thing as B7, just that B7 has two subtypes, B7-1, which is CD80, and then B7-2, which is CD86. All these are the same thing. Though I'll be honest, I was working through some questions and before I had known this, and I was able to answer the questions still just on the basis of being able to reason out what CD80 and CD86 could be. I thought they were like a B7-like molecule, right? But no, they're just different name for B7. So really, if you try to understand things, if something comes out of you from left field like that, and you can reason things out, maybe it works to your advantage. But it's not for me to tell you how to learn. You've come this far on your own after all, right? So what are these different flavors of helper T cells? You have Th1, Th2, Th17, Th0, and also you have follicular helper T cell. But we're only really going to talk about Th1, 2, and 17 today. So Th1 is going to be good against viruses or bacteria, although Th17 has some antibacterial function too. But then you have Th2, it's going to be your gastrointestinal pathogens or parasites. And then Th17, that's going to be versus your fungi or extracellular bacteria. And then you have something called Th0, it's kind of like a reservoir group. They're kind of like a jack of all trades. Just say that you have some Th0s on the side, right? And let's just say you have a response that needs Th17, which is fungal, and then you end up needing more Th17 than your body might have initially anticipated, it can shunt some of those Th0s to become Th17s, which is quite amazing that the body can really have this backup reservoir at the very molecular level. So yeah, so let's go into these and then work from there. So your Th1 helper cell. So again, we just have the whole 9 yards of the T-cell receptor on the cognate antigen, and the MHC matches up on the CD40 to CD40L and the B7 to CD28. You have all that. But then also the dendritic cell is going to smother this T-cell with interferon gamma and interleukin 12. Okay. Once this happens, the T-cell naive CD4 will become an effector cell, but it will become Th1, which is a type of an effector cell, or the CD4s, right? So what does Th1 do, right? What's the point of having this? Th1 is going to produce a very interferon gamma friendly type response. And so if you remember, like interferon gamma was our G for good morning because we're waking up macrophages, same story. So it makes quite a lot of sense that the dendritic cell suspects something bacterial. It's going to want it to have a macrophage friendly response because macrophage can eat bacteria, yeah? So that's exactly what the Th1 response is. It's a macrophage friendly response. It's very macrophage-esque, right? And so what else goes into that? Tumor necrosis factor, which also goes into activating macrophages, or hyperactivating rather. Also NK cells, right? Because maybe if there's some bacteria hiding inside cells, you want that means of killing as well, right? And interleukin 2, which is just a growth factor for T cells and NK cells, and this one's non-specific, right? So if you talk about clinically, because that's really how a lot of these things are presented, right? Or at least we try, right? So there's something called the granuloma. Granuloma really, don't confuse yourself. It's just really an aggregate of macrophages trying to wall in a pathogen. And so in tuberculosis, this really happens a lot, everywhere really, but big examples in the lungs. So if you see something about tuberculosis, you're going to think granuloma, you're going to think granuloma, you're going to think macrophages walling things in. You're going to think macrophages walling things in, you're going to think activated macrophages. You're going to think interferon gamma, you're going to think tumor necrosis factor, and you're also going to think Th1, because Th1 is the cytokine factory, if you will, that's going to produce interferon gamma, tumor necrosis factor, to direct a very macrophage-friendly response. So this is the big contrast also between CD8s and CD4s. CD8s directly kill problematic cells. CD4s don't. CD4s are cytokine factories that will in turn direct other white blood cells and also our baseline human physiology into acting a certain way to make a hostile environment for said pathogen, right? So this Th1 cell isn't going to directly go up to bacteria and kill it. No. It'll make this chemical environment that'll make other things kill it. And that's a key distinction. And that's why we call them the helper T cells, not killer T cells, as opposed to CD8 who can engage in direct cell killing, usually for like our own cells if there's like intracellular bacteria or virus hiding inside or something of that nature, yes? And then in terms of something like bacteria, typically cleared by macrophage, something like fungus, cleared by to some extent macrophage, but also neutrophil, huh? So these things are connected, but that's the thing, the helper T cell is a helper, it's not a direct killer, okay? So again, Th1 response, macrophage-esque, at loss of better words. Then you have Th2 cell, okay? This one is going to deal, well, we backtrack. Dendritic cell comes into lymph node, right? And he's going to do the presentation, right? And he's going to present maybe fragments of gastrointestinal parasite, right? And also IL-2 and IL-4. You can say, Khaled, well, what is IL-2? We saw IL-2 on the last slide. Well, again, I said IL-2 is non-specific. IL-2 is just the growth factor for NK cell and T cell in general. It's just a growth factor. So just appreciate the self-amplification. It's stimulated by IL-2, and then also it can promote other IL-2, right? So it promotes itself. It loves itself. Th2 here with the self-love in 2022. Congratulations. We love it. But also the IL-4 is going to stimulate. And the way I remember this is that IL-4 is like multiple of 2, right? So what does Th2 do? I hate how that rhymes. It's going to produce IL-4. And so the way I like to remember the function of IL-4, IL-4 is going to, remember how I said that I give like that thing that was like hot pot mnemonic or analogy for B cell isotype class switching? Yeah. So depending on the chemical environment directed by helper T cell, For the B cells, it can isotype switch into IgA, IgG, IgE, away from its default IgM form, right? And that chemical environment, that broth, remember the analogy I gave on hot pot, that B cells like the meat, and then the cytokine environment is the broth. The broth is actually produced by helper T cells in several regards. So if you want to make an IgE class switch, you want to cook the B cell in interleukin 4. And so there's mnemonics out there, but the way I like to remember it is like when you draw the letter E, you draw the letter E in 4 strokes. So IL-4 is going to make IgE response, right? So that's what that is. Okay. So also IL-5 is going to do promote to class switching to IgA. And the way I remember that the alimentary canal is 5 parts: esophagus, stomach, small intestine, large intestine, rectum, right? So, and then because it's like mucosal service, IgA, and very like GI oriented as well. So IL-5 mucosal switching to IgA, right? And again, IgA is the most abundant in the side antibody isotype in the body, as opposed to IgG is the most abundant just in the serum. But because again, remember, if we take our large intestine and spread it out like it's like the size of a tennis court. That's a lot of surface area. So you can imagine there's a lot of IgA there, right? You can also do IL-13 is going to stimulate the mucus production, and mucus is filled with things called mucins, which are these really large carbohydrate complexes that are really good at trapping pathogens. Yeah. Oh, I also want to make the note, because I did talk about the antibodies here when we spoke about our past for the what is it, the IgG, right, for Th1 response. Interferon gamma also promotes class switching to IgG3. Yeah, it's good I mentioned this. So why I mentioned this, IgG3 is actually one of the isotypes that's very adept against bacteria, because it's very good at fixing complement. So we spoke about last time 2 IgG or 1 IgM, because remember binding sites is good at fixing complement for like classical cascade, for example. Right. So Th1, the interferon gamma does multiple things. It wakes up macrophages, but then also it promotes complement deposition onto the pathogen. And what does complement do? Complement is direct cell killing, but also the deposition of C3b. C3b is also opsonin. So in addition to producing this interferon gamma friendly response, producing or waking up macrophages, it also makes it easier for macrophages to eat, because it's going to opsonize things for them, right? And even IgG3 itself is also an opsonin if the FC region is exposed, of course. And even if we're having NK cell oriented direction, IgG3 can also assist in the antibody-dependent cytotoxicity, right? ADCC, right? In which NK cell binds FC region of antibody is also one forms of like interaction that NK cell can kill something, right? And this is also specifically holds true for like viral infected cells, right? So I guess I appreciate all the self-amplification that goes on within the same response. So yeah, it's a good thing I didn't skip over that. It's good to know the antibody associations with the response. So again, Th1 macrophage friendly, IgG3, Th2 very intestinal. So you want to think IgA, and also you want to think IgE. And remember, IgE is the antibody that's like locked and loaded on like the mast cell, and also the basophil. And really, a basophil is just a circulating mast cell. That was aside the point. Yeah. So Th2, you want to think IgE, you want to think IgA, right? Th17 is an interesting one. So what's going to stimulate it? TGF beta, interleukin 1 and 6, and then interleukin 25. I think my camera thing blocks the upper left corner, right? But it just says IL-1 and 6, and then IL-23. Right. So I paired the 1 and the 6 together on the basis that 1 and 6 are just pyrogens. They just induce heat, right? And really, if you want to remember, it just comes up a lot. The way I remember it, when you draw like the number 1, I don't know, it looks kind of like a candle, right? And it looks like a little bit of wax is melting. and then you would draw the 6. 6 really looks like a flame, right? Doesn't it? So I think it's like, okay, heat response, right? So 1 and 6, right? That's just how I do it. And then again for IgE, 4 strokes for the IL-4 induction, right? Anyways, so what's the point of IL-17? It's also going to do some bacterial function, something shared trait with Th1, but also it's the mainstay of fungal infection. And so IL-17 is going to do neutrophilic recruitment, okay? And also IL-21. So the way I also remember is Th7, so you had Th1 and Th2, and those are small numbers, and then you jump to Th17, that's a big number. So also the interleukins for this one tend to be bigger numbers too, 17, 21, 23. 21 is also going to do BSL class switching to IgG3, which is important for opsonization. And so this is actually something it shares with the Th1 interferon gamma response. And also it's going to do some IgA as well, which shares with the Th2 response a little bit, right? And something good to, well, why IgA if we're thinking maybe fungal? You know, you can think about, I'll give an example. So sometimes when you're seen in women, just the basis of this, sometimes you're treating a bacterial infection, you're going to have antibiotic. And then antibiotic is good, but it kills all bacteria. So it kills good bacteria and bad bacteria, also better words. And so then your skin, or specifically really groin area, because it's very microbe-rich down there, is going to have a deficiency of bacterial microbes. But what persists? Fungi, because fungi aren't killed by antibiotic. You need antifungal to kill a fungus, not an antibiotic, right? So the fungus has no competition. So even though it's normal flora, but because it has no competition, it'll go rampant, and you get candidiasis, and vaginal candidiasis is a thing. So you can kind of appreciate why, you know, we have specific helper T-cell subset to keep fungi in check. Right. And so it's just good to know. And also, if a Th1 response is insufficient, sometimes it can induce a Th17 to try maybe to give it another go. So, but for basic purposes, Th17 response, fungal, and then neutrophils, right? Okay. And IL-23 is just a Th17 growth factor, kind of distinct from IL-2 just being the growth factor for Th1 and 2, but there's a 2 in 23, so that helps you a little bit. And so it's good to know that there's like feedback loops. And all that means is that, let's just say you have, let's say you get like a pinworm infection, which I think is Tuberculosis, right? Tuberculosis is a pinworm. It infects your GI. Well, I hope it doesn't infect your GI, but you know, it can infect the GI. So you're actually going to get a Th2 response. And so with the Th2 response, you're going to get your associated IL-4 and everything, right? All these cytokines of the Th2 response are going to suppress the other responses. So you're going to get less Th1 response, you get less Th17 response. And that's really what it is. And so that's really the immune system's way of hyper-focusing and directing the immune response to one direction. And that if you get one type of Th response, it's going to prefer itself, self-amplify itself, but then also suppress other ones. So just hyper-focused. And it's really good to get a type of immunological thinking. You know, the psychology of the white blood cell, because really, what more is immunology than the psychology of white blood cells? And you can meditate on that one. So I took the time right now to go into something called, and maybe this is why the first presentation took such a long time. Hopefully I can do it better this time. You have 4 types of hypersensitivities, and I'll do a whole lecture on them, but you know, they're quite nice and they're quite testable also. You have a type 1 hypersensitivity, right? That's just going to be very immediate, and that's going to usually be IgE mediated. So you can also appreciate it's going to be associated maybe sometimes with a Th2 response, because IgE, IL-4, right? This is like your bread and butter allergy. You have something called a Th2 response, and that's going to be an antibody attacking self-tissue. You have something called type 3 hypersensitivity. But I guess I backtrack in case I misspoke. It's a type 2 hypersensitivity, right? And then type 3 hypersensitivity is going to be immune complex formation. You have something called a type 4 hypersensitivity that's not antibody mediated. It's direct T-cell killing of something, or direct T-cell response. And the reason why it's called delayed type and it's kind of its own thing, even though it's very similar to type 1 in several regards, but the type 4 response takes a long time. And so the way you remember that there's 4 types of hypersensitivities, the biggest number, the longest number, takes the longest to do onset, and that can be like 48 hours, as opposed to the other that can kick in with only a few hours or immediately in some cases. So type 4 hypersensitivity, the basis of the immunology is quite interesting. 19th century, you had something called the Tuberculin skin test that was invented, and it's a PPD. So if you're like healthcare worker, or you're on your way to becoming a healthcare worker, before you go into like hospital setting, you usually have to get your tuberculosis testing done. So basically, they took this protein called tuberculin off Mycobacterium tuberculosis, which is an acid-fast rod, really tricky pathogen, and they purified it. So hence it's called the PPD, purified protein derivative test. So tuberculin is injected into the skin, okay? And then if the point of injection indurates, and that means gets hard, this indicates that your immune system has a history with tuberculosis exposure. And the basis of this test is that, you know, you get this Th1 response because it's bacterial in nature. Basically, you get the whole signaling, Th1 cells producing a very like macrophage-friendly response. And you get the interferon gamma tumor necrosis factor release at that site of the injection. And the immune system gets enraged at the site of the infection, or at the site of the injection. But then you could raise the question that if you have to do this test, you just injected tuberculin to someone, and let's say like healthcare worker, you have to get the test like every year or every few years. Well, you injected the person with tuberculin, so won't every test down the line be positive test? The answer is no, because it's a purified protein derivative, and tuberculin on the Mycobacterium tuberculosis isn't the danger signal. Tuberculin itself is on the bacteria, right? But it's not dangerous. Tuberculin itself is not dangerous. It's other antigens really that fire up our TLRs and everything. Tuberculin itself is quite harmless, and for this reason, it can be used on the basis of the test. How does this apply to anything? The thing is, so United States, for example, and other countries, tuberculosis is not as widespread. So this test can be done quite readily. However, in other countries where TB is quite more endemic, something called the BCG vaccine is given. And the issue about well, not really an issue because it's a good vaccine, but the thing about the BCG vaccine, it actually gives these tuberculin skin tests, makes them all positive. So usually for the, if you get tuberculosis testing in the United States, for example, and you are from a region or a country that gives BCG vaccine, you actually have to do a blood test. In the United States, the two main ones is a quantiferon gold and T-spot. Or you can just do it because you have the preference. It's a little bit more expensive, but for example, if you want to do tuberculin skin test, it's a delayed type hypersensitivity. You have to go to the health department twice. You have to get once to get injected, and then once like a day or two later for them to like clear you to see if it's indurated or not. As opposed to if you want to get the blood work done, it's like an assay. It's like an interferon gamma assay, I think. It's just one blood test. Like you just show up to the lab, you get your blood taken, and then that's it. So personally, I prefer the blood test as opposed to having to go to the health department twice, but subject to finances and insurance and so many other things, right? But yeah. But why would a positive test yield induration? Well, that's because if you're exposed at some point, your immune system recognizes it. So you have memory T-cells, and then they'll kind of like mature, and then so they've recognized tuberculosis antigens, the other ones, but they also recognize tuberculin, and so there's kind of like that association, you know. So There's also a thing you can say though, that it's not advised to do a tuberculin skin test when you're really sick, because it kind of goes back to white blood cell psychology. If you're sick, your immune system is enraged, right? If you inject tuberculin, even though tuberculin is harmless, because there's and you're sick for another reason, but because body detects something foreign while you're already in enraged state, it might associate the tuberculin with that sickness, even though it's not the cause. So sick people aren't advised to get TB skin tests, right? Because the danger signal is already up, right? But again, if induration forms, that means your body currently have latent, or you've previously had a TB infection, and that's the whole basis, because the T cells were even able to recognize and kind of like restart and yield this very sophisticated response. And again, because it's macrophage mediated Th1. We did this slide last time, but just how killer T cells kill. Mechanisms are shared with the NK cells. I won't harp on it too much, but just that's the difference. Again, CD4 cells are your type of cytokine factors, and then your CD8s are they directly kill infected cells. So they can perforate the membrane with perforin, literally cause apoptotic pathways to get triggered with granzyme B. Apoptotic pathways, caspase, the whole 9 yards, tidy cell death. There's also the suicide button of the body, which is FOS, and the T cells have CD8 cells have FOS ligand bind FOS, and if that interaction occurs, it also triggers apoptotic cascade. And again, NK cells share these with the T cell. So you have the two different. I did some patho review in here as well, pathophysiology review. Necrosis is usually going to be external damage, and it's going to usually be it's very untidy. It's going to have free radicals and toxins and everything. It's usually an untidy affair. And you have apoptosis or apoptosis if you're one of those. It tends to be a more controlled cell death. And then right now they're discovering subtypes of apoptosis, and then, oh my God, they did it to me. You have subtypes of apoptosis, and some even are like an intermediate between apoptosis and necrosis. That's really beyond what we're going to talk about today. But just generally know apoptosis tends to be a more tidy affair. Right. And then when we talk about types of necrosis, there's multiple types, right? Well, what do all types of necrosis have in common? There's an exogenous insult and local inflammation. And now how the tissue dies, necrosis is tissue death, it can vary. So if you have a solid organ, you're going to get, usually the death is going to be secondary to ischemia or an infarct, no blood supply. You get coagulative necrosis. Very similar mechanism exists in the brain, but because the brain is a soft tissue, it's going to go liquefactive necrosis. And you can think of something as soft as more liquidy. It melts, your brain melts, you're watching this lecture and your brain melts. Not my intention, but I hope I'm not overwhelming anyone. You also have caseating necrosis. Usually it's secondary tuberculosis, which I'm sitting inside. We'll talk about the granuloma later. This is my background. I'm in a granuloma right now. But again, you can appreciate that that's a Th1 response because macrophages are walling up the pathogen. So caseating necrosis really associated with TB. You have something called fat necrosis, a classic scenario that you get some kind of something in your peritoneum, right? Goes ARI, and pancreatic lipases spill out, and they can melt the fats. It's called saponification. That's a fat necrosis because lipases melt fat, right? And so that kills the fat cells. So that's also another one is take a direct hit to breast tissue because very rich in fat, and the trauma can actually cause death, right? Blunt trauma can cause cell death, and that's also fat necrosis if it's hitting the breast, for example, right? Yeah, fibroid necrosis tends to be associated more with immune complex deposition, and we can talk more about that when we talk about hypersensitivities. And you also have gangrenous necrosis, usually it's usually ischemic, and it's affect usually the extremities, fingers, toes. It can be dry or it can be wet, and if it's wet, that means it's a liquefactive necrosis coupled to a coagulative necrosis. So it's kind of two things on each other. And the big example is this gram-positive bacteria. It's called Clostridium perfringens. So the Clostridia are this genus of bacteria. They're really quite dreadful. They are gram-positive, right? They are bacilli or rods, and they They form spores. That means they're very hardy. They can go dormant when the environment is not the best. And when the environment gets good again, the spore can actually bloom into a fully mature bacteria. And it's usually the classic thing for Clostridium perfringens. You get like a motorcycle accident, and then like the motorcyclist, he like scrapes a lot of his skin, and he's like up against the soil. And in the soil, there's Clostridium perfringens spores. They enter, and then the body is nice and warm and full of nutrient-rich blood and everything. So these spores like hatch into Clostridium perfringens, and then the Clostridium perfringens releases an exotoxin. This exotoxin is going to really mediate the formation of this gas gangrene. And they look like these dark blue purple bubbles like blooming out of dead skin. It's really an atrocious sight. You're free to Google it if you're into that. And so yeah, just to wrap up, that's really my background here. To drive home the point of like the Th1. So this is the granuloma of tuberculosis. This is inside a lung. And so this is called caseating because it's cheese-like. So in this bottom right-hand corner, you see this macro view. It's cheese-like. That don't mean to ruin your appetite. I'm actually fasting for the holy month of Ramadan right now. So it's, for me, it's good not to have an appetite. But if you're not in that position, then I'm sorry. I don't want to ruin cheese for you. But yeah, that's really how it gets its name, because it's like cheese-like necrosis. So on the micro view, though, just to look at some histopath, this is called the necrotizing granuloma. So granulomas can be of two types. They can be necrotizing and non-necrotizing, right? The basis of the necrotizing granuloma is that there's this central region of cell death. And so how do you know it's the central region of cell death? One, it's very eosinophilic, and then two, there's no nuclei. So all this region is dead cells. There's such thing as non-necrotizing granuloma. And you see that like in sarcoidosis. It would look very similar. It's like this, oh, to be clear, this whole thing of the granuloma, right? But then you get like a granuloma and non-necrotizing, like sarcoidosis. It would look like this too, like the circle amidst the lung tissue. But then in the middle, you'd still have nucleated cells, right? But this is necrotizing because in the middle of the granuloma, it's necrotic tissue. There is no nuclei, and it's like this big eosinophilic mass or morph goo, if you will, right? Something else you can see in this necrotizing granuloma, pulmonary tuberculosis, is at the periphery, you get these elongated cells, and these are actually macrophages, and these are called epithelioid cells. So basically, I said before, it's a Th1 response, very macrophage-esque. Th1 cells are going to promote a macrophage response, and these macrophages will actually go in and they'll try to wall off Mycobacterium tuberculosis, and they wall it off by becoming like a type of epithelial structure, right? And so that's what they circle. Additionally, in this necrotizing granuloma, I like this picture a lot because it's very clear. You see this cell right here? This is called the Langhans giant cell. And the way I like to remember it is that it's, well, first of all, how do you know? Look at this horseshoe-shaped nucleus. And for me, the horseshoe kind of looks like an L. Not really, but that's just how my brain works. And so in histology, there's like a phenomenon in which when you have a big aggregate of hyperactive macrophages, they can actually fuse. And so that's what a giant cell is. And this specific pattern is called the Langhans giant cell, indicated by horseshoe nucleus. And again, because we have the Th1 response going on in granuloma, because it's macrophage-friendly response, makes sense that the Langhans giant cell is an aggregate of macrophages, right? So it all comes full circle. And then usually if you have like these tadpole cells out in the periphery, I call them tadpoles, just these big nuclei, scanty cytoplasm. This usually means lymphocyte, but on a simple H&E stain, which we have right here, you cannot distinguish between a B cell, T cell, NK cell. Or it's very hard to do for NK cell, but generally B cell, T cell, you cannot distinguish. You need more advanced staining. You want to look at the surface markers, like immunofluorescence, for example. Like I think for B cells, you want to look at CD19, 20, 21. I think those are the B cell markers. But that's aside the point. But if we were to make a hypothesis, to make a bet, I'm not advocating for betting, or if we made a guess and say like what type of lymphocytes are in this lymphocytic infiltrate, they're probably Th1 because they're directing the macrophage response to begin with, right? So I thought we can just go into some histopath because it's a really clever way on the part of test takers to try to correlate like histopathology to immunology. You see macrophage-friendly response. You want to think interferon gamma. You want to think tumor necrosis factor alpha. You want to think Th1 response, right? So that's all for today. Hope I didn't overwhelm anyone. I hope I didn't go for like an hour in this lecture. We did talk about a lot though. So I guess in that regard, I hope it's useful. Thank you for your time and your attention. Next time we'll talk about secondary lymph organs. And here are my sources. Take care, everyone. Take care.